So I'm pleased to be hosting Dr. Elisa Crisci to discuss her recent PLOS ONE Lab Protocol paper entitled High-throughput 96-well plate-based porcine antibody isolation protocol and the associated detailed protocol in protocols.io. Elisa is an assistant professor at North Carolina State Veterinary College. She studied veterinary medicine at the University of Bologna in Italy and earned her master's in science and doctoral degrees at the Research Center for Animal Health, or CRESA, in Spain, where she studied virus-like particles as vaccine vectors for animal viral diseases and animal host-virus interactions. After completing her PhD, Elisa spent further time at CRESA using pigs as a large animal model for immune cell therapy, performing in vivo dendritic cell tracking. After Spain, she took her research to the Linköping University in Sweden and moved from pigs to humans, investigating the interaction between human viruses, innate immune cells, and the complement system. And then back to pig viruses at the French National Institute for Agriculture, Food, and the Environment. Elisa joined the faculty at the College of Veterinary Medicine of North Carolina State University as an assistant professor in virology. She defines herself as a swine viral immunologist, and her research addresses virus-host interactions in porcine diseases with a focus on influenza virus and other viruses. Elisa, thank you so much for talking to me today.
Thank you very much for the invitation, for being here.
So to kick off, can you briefly explain what the protocol is about, how to use it, and why it is relevant?
Sure. So what we did is to develop a high-throughput method to isolate pig antibodies. And we started doing this because we encountered some issues in performing ELISA in a recent project we were working on, and was specifically testing maternal antibodies transfer from lactating and pregnant sows to piglets. So when we were working on that project, we realized that particularly in sows that are pregnant and lactating, their antibodies are a little bit sticky. So sometimes we were having issue in a high background or in a specific binding when we were performing our ELISA. And so we started to isolate the antibody, but initially that isolation method was based on single column chromatography. So we're very, very burdensome in terms of time, and try to isolate more than 200 samples at a time from all our animal studies was pretty heavy in terms of time and commitment. So my technician, that actually Jake is the main, leading person for this protocol, started to develop a high throughput based system to actually isolate pig antibodies. And in particularly, we were able to isolate IgG and IgA isotype. So he was able to actually run 192 samples in few hours instead to run like 6 of 12 in a few days. So that was helping a lot, the productivity and all the downstream assay that we were using with those antibodies. I want to point out that we didn't have any pig system, really. We have to adapt all the resins that we use to isolate that antibodies for the pigs. So those resins were generally described for other species, particularly human, rodents, but they were not really described for pigs. So we were trying to actually adapt that kind of isolation system for our species.
And I wonder whether, the focus of, or the reasons for optimization of the protocol was to speed it up and to optimize the time. I also wonder whether there is a cost associated, and you actually reduce by replacing the columns with 96 well plates, whether you reduce the cost of what was known at the time for antibody purification. Is that correct?
Is that correct? So we decreased the cost, the time, but we also, that was the only system actually that allowed us to see proper differences. For example, this project was working on influenza infection, so we start to encounter problem because there was this in a specific binding to our hemagglutinin protein that we use for our protocol. So without that isolation, we were not seeing, for example, differences between vaccinated animal and unvaccinated because of the inespecific binding was so high. So we needed to isolate the antibody to really see differences between the so the vaccinated animal, so the specific vaccine response against a specific hemagglutinin versus the unvaccinated animal control animal that were still showing some background level, but that disappear after we isolate the antibody. So it was at the same time a reduction of cost, but also we were able actually to see significant differences between our vaccinated and unvaccinated animal because of their particular status of pregnant and lactating sows.
And building on that, can you elaborate on the sort of experiments or applications that this protocol, the high throughput, the higher affinity or higher specificity will enable that weren't possible before?
Sure. So just imagine before we were using in particularly three main type of samples like serum, milk, and colostrum. So milk and colostrum are very complex samples with as a matrix is very complex. So when we were using those samples, even with dilution, we were not seeing any specific antibody against, for example, hemagglutinin in our in-house ELISAs. After we apply the isolation method, we were even being able to differentiate IgG specific and IgA responses towards specific hemagglutinin. We could see differences in specific antibody responses and even specific isotype between, for example, vaccinated animal against control animals. Of course, this cannot be applied to other biological fluids, but these were the ones that were much more complicated and that were giving us the highest background in our immunoassays. So that's why we focused the protocol on this type of samples.
I would like us now to go a little bit more into the development per se of the protocol. And I don't know if you can share with us what were the highs and lows during the process of optimizing the protocol? And was there anything that you tried that didn't work and didn't make it to the final version of the protocol?
So for sure, the highs, it was that we were able to, isolate 200 sample at a time almost, and optimize the time management for working on the project, in particularly for my technician. So that was the most positive, the high of the protocol. The low is everything related to standardization and validation of the protocol. So we had to compare the single column versus the plate base. We have to, for example, refine the resin concentration, refine even the sample input, and try to find, the best combination of those different aspects to have the best protocol at the end. As every protocol, there are a lot of standardization going on, troubleshooting. So it took a while. We knew that we have resin that we could use in pigs, but when we started, they were not described for pigs. So everything was, troubleshooting the concentration and the amount and try to adapt to our samples.
As you were developing the protocol and standardizing it, was there anything that you thought that should have worked, that you thought based on your intuition, but at the end, when you actually performed the experiment, that was wrong and you had to do something else?
So at the beginning, we were not doing isolation because there are so many papers that actually use all samples in pigs, in particular for serum. Not that much for milk and colostrum, but for serum for sure. but most of the time those were referring to growing pigs, so different age of pigs. While when we started to work with pregnant animal and lactating animal, we figured out also based on human literature that specific status of the animal increase, for example, sialylation of the antibodies. And that change, of course, the binding capacity of antibodies, making them much more prompt to having a specific binding when there are free sialic acid. Of course, if you work with influenza, the main receptor are sialic acid. So we've been working with a virus that needs that kind of receptor. So everything was complicated to the fact that we were using influenza for our project. And we had a very particular status of the animal that were increasing sialylation. So that's why we had to actually move forward and start to do the isolation because of that problem at the beginning when we started work with these pregnant animals.
If the listeners would like to bring their protocols to their own labs, what is your advice in terms of the most critical steps and any parts that might be particularly tricky and others might make a mistake on?
Yeah, for sure. The resin concentration will need something you need to work on or try to standardize. It should be pretty easy to use the one we have been using, but that's something that you need to work. And it will depend also what the type of samples you're using and what species you're using. If you work with rodents, there are, for example, other protocol that you might be able to use for this kind of isolation, or you can just adopt our protocol because our resins can actually be used in other species too. So it will mainly just, do some troubleshooting on the sample that they have and the amount, mainly the amount they need to use for their input and the resins that they are using for isolations.
I would like to change gears now a little bit and explore your motivations to send the protocol as a standalone methodological paper to PLOS ONE to be peer-reviewed.
Yes. So we wanted to do that because we figured out that was actually pretty unique in its type. There were not that much work in the pig scientific community. And we thought that was actually relevant for the scientific community to have a protocol to use for this antibody isolation. And we know that this protocol is pretty versatile, can be used in different contexts for antibody isolation. So that's why we wanted to, send it as a standalone, and PLOS was one of the best choice for this kind of protocol. Also because our institution has a particular relationship with PLOS ONE, and that's why we wanted to use that kind of agreement for our publication.
How did the feedback that you received from the peer reviewers shape the final version of the protocol?
I have to admit that the reviewers were very positive, and we found their comments very helpful to improve the quality of the paper and also to make it more accessible to the general public. So at least in our case, our reviewer experience was very positive, and we received very positive comment and very useful for the paper revision.
Why did you decide to make the peer review history public together with the paper?
Well, I don't know. Personally speaking, I like for everyone to see how the process goes and what are, sometimes you have also additional questions that we answer that are not reflected in the paper, and that might be useful for the community, for the scientific community, for other studies. So I thought was actually pretty useful to have that open.
So my last question might be at this point a little bit speculative because your paper is relatively young. It was published less than a month ago. But what do you think will be, if not has been, the significance of publishing this lab protocol paper for your career and for those more junior staff at your lab?
So the main significance, this was the first first article lab protocol of my technician in my lab. So it for him was a very good professional development. Also, he was writing and leading this project by himself. And from my perspective, we are going to use this protocol in this paper in other paper that we are planning to submit soon that are part of this big project we were working on in influenza and maternal transfer. So from my perspective, this was a useful publication that we are going actually to use for other publication in the future.
Elisa, I don't have any other questions. I don't know if there's anything that you would like to add that hasn't been touched so far in the conversation.
I don't think so. I mean, I think I've described the majority of our outcomes, and we're actually pretty happy about this publication, this protocol, and I hope actually other pig researchers will be able to use it for their studies, and I hope it's helpful for them.
Thank you so much.
Thank you.